Test, test. Okay. All right, can you get a sound check? Sound check in the back? Everyone hear me? Thumbs up, around, looking in the back. All right. So, hey everyone, welcome. Thursday. Wanted to change it up this week for kind of the check-in question. Usually it's kind of just checking in yourself right now, but sometimes it helps to kind of look forward. What's one thing you're going to do for yourself this weekend? Let's see. Where is everyone feeling? What's everyone thinking? Nap? I can 100% appreciate that. Check in with a friend, drink some water, get outside. Hopefully it's going to be not too rainy and no snow. Go to bed early. Yep. Exercise, meditate, stretch, paint, draw, dance. Wonderful. Read for fun, watch a movie. Curiously, no one's stepping away from screens. That's fine. Eat healthy, not necessarily there as well. Oh, one popped up. So eating healthy, maybe trying to get back to some routine if you've been a little bit stretched and maybe just more in that survival mode right now instead of kind of planning ahead. I know for me, I am going to go with stepping away from screens. Someone just popped up there as well. My phone broke a couple days ago, so I've had to kind of reset everything, but this might be an opportunity for me to decide maybe what I actually want on my phone and if it's going to be more of a distraction or more something useful. So I'm going to go with step away from screens if I can. Awesome. Thank you for sharing. So this is your last official lecture with me. I think maybe you'll see me again towards the end of at the end of the term as we get ready for the final exam. But this is kind of going to culminate what we've been talking about over the last couple of weeks. We are at the end of the line for our carbohydrates. We're at the electron transport chain. Through these last couple of weeks, we've looked at carbohydrates and their structure. We've looked at what makes them unique, what makes them complex, and why they're important. We've seen how they're moved around within a body, through the blood, across cell membranes, and into the cells where they can be used for energy. We've seen how they can be broken down and metabolized from that 6-carbon unit to 2 3-carbon units in pyruvate. We've seen how that excess glucose can be stored as glycogen and then broken down again. We've seen what happens to pyruvate after glycolysis, either when there is oxygen or mitochondria available, or if there's not. We've followed it through that into the mitochondria, into the through the TCA cycle to see where it's getting oxidized to carbon dioxide, burning it to release that potential energy. And now we have a whole bunch of NADH molecules, and we're going to see what's happening to those NADH molecules today in the electron transport chain. But through all this, we've also seen how regulation is important. So I want you to keep that in mind because it's going to feed into our discussion today as well, and you're going to start to see how regulation and understanding of regulation can help us understand how we use metabolic fuels. So for today, I want you to focus on, or I want you to pull out of this lecture how the electrons flow through the electron transport chain and generate that proton gradient, that electrochemical gradient that is going to fuel ATP synthesis. I want you to examine the conformational changes, so the changes in protein structure that occur with ATP synthase as it generates ATP, or through oxidative phosphorylation by bringing an ADP and a phosphate together to form that ATP. I want you to define the PO ratio, or the phosphate to oxygen ratio, for NADH and FADH too. These are how many ATP molecules can be generated for every oxygen molecule that's going to be reduced. And then to be able to calculate the number of ATPs and water molecules that we produce from a single glucose molecule. And then you should be able to apply this for the other metabolic fuels you're going to look at later on over the next couple of weeks with Dr. Patterson. So keep this in mind as we move through this course, or through this lecture, and as you prepare for the final exam. So like I said, we've come to the end of the line for our carbohydrates. In the TCA cycle, those carbohydrates now have been fully oxidized. That 6 carbons of glucose has resulted in 6 CO2 molecules. And the energy that was stored within those chemical bonds has been transferred in some part to ATP, but most of that energy is now held within these NADH molecules, the potential energy related to these electrons that are stored there. In order to extract this potential energy, these electrons need to be passed from NADH eventually to oxygen. This movement is going to be used to fuel those conformational changes in ATP synthase, allowing the movement of protons across that plasma membrane, generating that proton gradient. And then ATP synthase is going to use that proton gradient to couple the movement of protons back into the mitochondrial matrix to generate ATP. Now this is all about energy transfer. The energy that's stored, or the potential energy stored where in NADH with those electrons, is going to be used to fuel or to cause the conformational change in these proteins, allowing us to move protons from a relatively low concentration to a relatively high concentration against its concentration gradient. And remember, the first law of thermodynamics, energy can't be created or destroyed, it can only change form. So some of that energy is used to fuel this conformational change. However, not all of that energy is used in this way. We lose some of that energy as well. Unlike within the glycolysis and the TCA cycle, where all of that energy in those chemical bonds are essentially captured within ATP or NADH, here, not all of that energy is used for that transfer. Some of it's going to be lost. And since energy can't be created or destroyed, it can only change form. In this case, some of that energy that's lost converts into heat. And this is actually how we as humans maintain our body temperature. The metabolic processes maintain the temperature in our body, keeping it at around 37 degrees Celsius. And then during times when our body temperature increases, maybe you're sick, maybe you have a fever, that signals to your brain, your hypothalamus, to start to regulate your body temperature, to increase your body temperature, to make it a more inhospitable region for bacteria or pathogens to grow. And the way it does that, by raising your body temperature, activating these metabolic processes, activating the electron transport chain, burning fuel for energy to raise your body temperature. So this is an important feature in how we regulate our own body temperature as well. So we do lose some of this energy as heat, but that it becomes functional for us and physiologically important for us as humans and other warm-blooded animals. But that proton gradient is what's fueling this. So we're using some of that energy to move protons across that plasma membrane, and that now concentration gradient is what's being used to generate ATP in the other direction. So looking at this electron transport chain, you may remember this from high school, from other courses as well. So we're going to review it very briefly, but these are the key important features. Those NADH molecules have those electrons. Their potential energy in the electrons they're storing. Those NADH molecules are going to donate those electrons or transfer those electrons through a redox reaction to complex 1, generating NAD plus, and then two hydrogens. As those electrons go through the complex 1, these are a series of redox reactions where those electrons are going to move from one molecule to the next. Through complex 1, they're going to follow and move and be transferred to coenzyme Q in another redox reaction. I'm going to just change this to a different color. Now those reactions that are happening within complex 1 are going to cause the protein to change its shape. And this facilitates the movement of protons from one side of the membrane across to the other side of the membrane, pushing those protons across. From coenzyme Q, another redox reaction is going to occur at complex 3, causing the same or similar conformational changes. Those conformational changes will take protons from one side and help push them across the membrane to the other side against their concentration gradient. From cytochrome C, they move to complex 4, where eventually those electrons are going to make their way to oxygen, generating a water molecule. This complex also is going to translocate those protons across that membrane. In total, we see complex 1 is going to transport both 4 protons, complex 3, another 4, and complex 4, 2 protons. So from NADH, we're going to put 10 protons across the plasma membrane. Now FADH2, cannot use complex 1. Remember, this is a redox reaction, this is a substrate. Complex 1 requires NADH as a substrate. Complex 2 is going to use FADH. In fact, it has an FADH molecule in it. Remember, complex 2 is succinate dehydrogenase. This is part of the TCA cycle. So during that step within the TCA cycle where we're generating an FADH, this is where it is. It's already part of complex 2. That FAD is going to be released to generate FAD total, transferring those electrons through the electron transport chain. Going from complex 2, and complex 2 does not transport protons across that membrane. It only carries out a redox reaction to transport or to transfer those electrons over to coenzyme Q. From there, it moves through complex 3, just like with NADH, coenzyme Q, and eventually finding their way over to oxygen. Because the electrons in FADH bypass complex 1, they're only going to transport protons across at complex 3 and 4. So that means we only have 6 protons transferred for every FADH molecule. Now, those protons that are on the other side of the plasma membrane, they are going to have an impact. We see the concentration of protons increases in the intermembranous space relative to the matrix, which has a relatively low proton concentration. But we've seen proton concentration before in this course. What does it mean when something has a very high proton concentration or a very low proton concentration, or relative differences? How do we measure that? You can shout it out. pH. So what is going to be the relative pH in the intermembranous space? Is it going to be relatively high or relatively low? High protons, low pH, right? On the other side, low concentration of protons, that pH should be relatively higher. So we're going to see the difference in the pH of these environments and how it has an impact on some of these processes. But this relatively high concentration of protons now in intramembranous space is going to fuel the molecular motor ATP synthase. Those protons can now move through ATP synthase, causing that conformational change. And that conformational change is what's going to bring ADP and phosphates together to create that ATP molecule, snapping them together. So we're not transferring the phosphate from a substrate. So this is not substrate level phosphorylation. We're transferring it, we're bringing them together in what we're calling oxidative phosphorylation. Now this is an animation that Dr. Patterson generated. So just seeing that flow kind of in real time. Seeing NADH transferring its electrons to complex 1 through that chain, eventually to complex 4 and to oxygen. And that's coupled with the movement of protons. FADH does the same thing, going from complex 2 to 3 to 4, combined with the flowing movement of those protons across that membrane. And that ATP synthase is where those protons come in. So as long as we have that proton gradient, ATP synthase can generate energy, generate ATP. But if anything disrupts that proton gradient and we no longer have that difference, we're unable to generate ATP anymore. So these proteins within the complex have to be able to move those protons across the membrane. So looking at the thermodynamics of this, the energy generation, we can see changes in potential energy as these electrons are transferred. Often you're going to see transfer from something that has a very high energy, transferring to something that has relatively lower energy, or moving across that. Like we saw within our cycles, moving from a relatively high energy to a low energy, releasing that energy, allowing these conformational changes to occur. Looking at this process, again, from this perspective, we can see when NADH moves through this process, this chain, transferring protons across the plasma membrane, we're going to see changes in what we call potential energy, or standard reduction potential. And we're going to talk about that in a couple more slides. When complex 2, or succinate dehydrogenase, transfers the electrons into that pathway, it also is going to result in changes in energy, that free energy that's going to be used to fuel these conformational changes in these processes. So looking at the path of these electrons, they're going to go from something that's going to be has higher potential energy, NADH, to something that has relatively lower potential energy, or is relatively stable, oxygen. So looking at the standard reduction potential, this is really just speaking about a molecule's tendency to be oxidized or reduced. Remember the acronym? Leo the lion goes grr. So loss of electrons is going to be oxidized, gain of electrons is going to be reduced. We can use this delta E, or this standard reduction potential, to determine if a molecule is more likely to be oxidized or more likely to be reduced. If it has a very negative delta E, or standard reduction potential, it's going to be more easily to lose its electrons and become oxidized. Something that has a very low or very positive standard reduction potential, this is going to be more likely to gain electrons and become reduced. So we're able to use this to track the movement of electrons within our pathway. Now this standard reduction potential is related to our other thermodynamic constant, the Gibbs free energy. Gibbs free energy is going to be proportional to the standard reduction potential. If we look at this formula, if we look at this formula, N here is going to relate to the number of electrons. F is Faraday's constant, which is 96,485 joules per volt mole. And then we have our standard reduction potential. What does it mean, or do you remember what it means when Gibbs free energy is going to be negative? Is that reaction going to be favorable or unfavorable? Favorable, right? If it's positive, it's going to be unfavorable as written. Excuse my terrible penmanship. So looking at this reaction, in order to get a negative delta G, because of the negative sign in this reaction, that means we need to have a positive change in reduction potential, and vice versa. For something to be favorable, we want to be able to see a positive change, or that change in that standard reduction potential as it moves through that process. So looking at the movement within our electron transport chain, the first molecule is going to be the oxidation of NADH. It's going to lose its electrons. The final electron acceptor is going to be oxygen, which is going to become reduced to H2O. So looking at the terminal ends, we can see the total change in standard reduction potential and calculate it. So when we combine these reactions, looking at what's happening in the electron transport chain, half molecule of oxygen, or just one oxygen atom, is going to accept the electrons from NADH with its proton itself to generate H2O and that now oxidized NAD plus molecule. So can you calculate the standard reduction potential of this? Looking as written, and we write these, and just as a note, we write these standard reduction potentials in this direction where a molecule is becoming reduced. So if it's happening in the opposite direction, you would have to flip it, right? Same thing we do with when we're looking at or chemical reactions. When we're looking in the opposite direction, we change the sign on Gibbs free energy. We do the same over here. So what do you see within the standard reduction potential? Sorry, yeah. What do you see within the standard reduction potential? Survey says, oh, there we go. Majority is going with 1.14 positive, which would be correct. So let's look at that. In order to look at the change, we're looking at the change from the acceptor to the donor. So we're looking at the change of potential as written here to determine the difference between these two. Remember, something that's going to be very negative is going to be more likely to lose its electrons. Something that's more positive is going to be more likely to gain its electrons. So when we look at the difference between them, the acceptor minus the donor, we get a total change of 1.14. Now for myself, the easiest way for me to rationalize this or visualize this is to use a number line. I've been teaching my daughter or helping my daughter with math lately, so we've been using a lot of number lines. Plotting this on a number line, 0 in the middle, we can plot both of these standard reduction potentials on either side. So for example here would be negative 0.32. We put the positive 0.82. The difference between those is going to be 1.14. So we're looking at the change in standard reduction potential between these two molecules, the difference between them. And that tells us how much energy is going to be lost with the movement of this, how much potential energy is going to be used or lost with the movement of these electrons. So looking at these, the following redox potentials for these oxidation reduction reactions, can you predict the flow of electrons if all of these were happening in sequence? How would the electrons flow, or which would be the first reaction that would flow to the next reaction and to the next reaction? How would these electrons flow? Would it go in which order? So which would be the first reaction, which would be the second reaction, which would be the third reaction, which would be the fourth reaction? So looking at the responses, there's two that are kind of coming to the forefront. A, so going from B to C to D to A, or small group of people also thinking it's going from A to D to C to B. So essentially these are flipped and reverse reactions. So I'll tell you now that the correct answer is A. Let's take a look at that on the back on the slides. Looking at the standard reduction potential, we are always going to try and move from negative to positive. So the most negative standard reduction potential here is ferredoxin becoming reduced. Next is glutathione becoming reduced, followed by ubiquinone, and then finally the cytochrome C. So we're going to see it going from B to C to D back to A. Using our number line analogy again, we can place these all on a number line and see the direction of electron flow. So going to 0.43, negative 0.23, then positive 0.1, and then positive 0.22. Since electrons are going to move from something that's going to be more negative to more positive, or moving from something that has a very high or negative standard reduction potential, we can see the movement of electrons moving towards the right from that most negative one to the most positive one, flowing across. And this is what's happening in our electron transport chain as well. So in the electron transport chain, those electrons are flowing from the molecule that has the most negative standard reduction potential, NADH, eventually finding their way to oxygen, causing it to be reduced to water, and releasing that energy along the way. By doing it stepwise, we can capture that energy to be able to fuel these conformational changes. If the electrons are transferred just from NADH just to oxygen, that's going to release a lot of energy, and it's difficult for the protein to be able to capture that energy and be able to use it effectively in that process. By doing this stepwise along this electron transport chain, some of that energy gets captured at each step to fuel the conformational changes within the protein to allow all those protons to be transported across the membrane. And eventually, that energy now that's been used to pump those protons across the membrane can be now used by ATP synthase. That potential energy now in that proton gradient can be used to fuel the conformational changes within ATP synthase to generate ATP. So how does ATP synthase make ATP from this proton gradient? And this is the culmination of Peter Mitchell's chemiosmotic hypothesis, is that electrochemical gradient, or the proton gradient across the membrane, is what's fueling the changes, the movement of protons back across the membrane is what's fueling these conformational changes, allowing ATP synthase to generate ATP. Now, ATP synthase is the key here. It's a membrane-bound protein, so it's going to have this transporter function, but also this enzymatic function. It's coupling these two processes together to allow this to occur. Looking at ATP synthase, it is, pretty sure, the first example of a molecular motor we've seen within the course. A molecular motor is something that's going to be able to move, rotate, and actually carry out an activity, similar to what you might imagine as an engine or a motor within the context of a car. That movement of those parts, in here, in case those proteins, is going to cause changes and cause changes within the conformation, changes within how molecules interact within those proteins, within those active sites, allowing these chemical reactions to occur. All the actual enzymatic synthesis, or the generation of ATP, is going to occur in what we call the F1 domain. So this is going to be the portion of the protein that's going to be in the mitochondrial matrix. It's going to be interacting with free phosphate, free ADP, and bringing them together and carrying out that enzymatic reaction. What we refer to as F0, this is going to be the portion that's embedded within the membrane. Thinking about this portion, this is going to be what's transporting those protons across the membrane. This is the beginning of the transport portion. So when a proton, here I'm going to just indicate matrix, inner membranous space, or intermembranous space. Looking at the protons, there's going to be lots of them on this side. As those protons move across, it's going to fuel the rotation of this rotor. We call it a rotor because it's going to rotate around. So as those protons move across, it's spinning the rotor, and that rotor is embedded within the beta domains here within this complex. As they rotate around, they're going to cause a conformational change based on the amino acids they're interacting with. That conformational change in the protein caused by that rotation has that ripple effect in changing the interactions of those amino acids here now in this portion, the catalytic portion of this protein. That's going to bring ADP and phosphate together, allowing it to catalyze that reaction and bring it together, forming ATP. So it's forcing them together, generating this molecule, this new ATP molecule that can be used for energy. Now each one of these, or the beta subunits here, there's 6 beta subunits, but they're going to work in tandem, so we have 3 catalytic active sites. They're going to go through a series of conformational changes, either being open, where ATP is going to be released. They're going to be in this loose conformation, which is going to facilitate ADP or phosphate binding, or in its tight conformation, which is going to bring those together and snap those 2 molecules together to form ATP. As the rotation occurs and the conformational change occurs, as that rotor rotates, you start to see each one of these subunits change its conformation sequentially, allowing these to snap together in this process to occur. So I'm just going to erase it here. Looking at these conformational changes on the right here, kind of just seeing the sequence. In that open conformation, it's going to be loose or open, and ATP is going to be able to leave. In that loose conformation, L, you're going to see ADP and phosphate being able to bind, and then it's snapping together in its tight conformation, forming ATP. So as they rotate across, a series of conformational changes will occur. This video kind of shows it really nicely. Looking at the structure, as protons move across that membrane, it's causing that conformational change in that protein. That conformational change is snapping ADP and phosphate together to form that free phosphate molecule you're seeing here in kind of purple, allowing it to generate that fully functional high energy molecule. Now this process is truly what we call a molecular motor. This rotation here is causing those conformational changes, almost like what you'd be seeing in an engine, generating that fuel that can be used by other cells. So looking at oxidative phosphorylation, we can develop an understanding of how these processes work, often by breaking it. A strategy we commonly see within, or we commonly use as biochemists, as scientists, is if we want to understand how something works, we want to break it to see the consequences of those reactions. Think of it more as a what if. Trying to understand how complex 1 works, we could say, what if we break complex 1? What is going to be the impact on ATP generation? What's going to be the impact on ATP synthesis? So if we can break sequentially each step in this reaction, we can gain a better understanding of how it works. And in order to break it or to stop those proteins from working, often we're going to use inhibitors. So we have a series of inhibitors of all these different complexes that allow us to understand how ATP generation is working within this electron transport chain and how to evaluate the differences between them. So for complex 1, we have rotenone and amytal. So rotenone is an insecticide, amytal is a barbiturate. These are going to stop the ability of complex 1 to transport electrons to coenzyme Q. That means we'll turn off complex 1. Now, if we turn off complex 1, are we going to completely stop ATP generation, or are we going to just weaken it? Show of hands, completely stop our ability to generate ATP. Show of hands if we think it's going to just weaken it. Yep, weaken it. Why? Yep. Yeah, so we still have the ability to allow electrons to flow through complex 2. Remember, complex 2 is going to allow FADH to flow. So we're going to generate less ATP because that's going to generate a weaker proton gradient, but we can still generate ATP. So by inhibiting complex 1, we gain a better understanding of how this electron transport chain works. Likewise, if we inhibit complex 3, so we could use antimycin A, we are going to have an impact on our ability to generate energy. So if we block complex 3 only, we'll have the initial transport of electrons from NADH through complex 1, pumping some protons, but we're not going to be able to use oxygen. Those electrons from that NADH molecule and from FADH are not going to make their way to oxygen. So we can see what impact that is going to have on the ability to generate ATP. Complex 4, we can be inhibited using cyanide, azide, carbon monoxide. So you may have heard carbon monoxide is inhibiting oxygen transport by binding to hemoglobin. It also binds to other heme-containing molecules as well, and we have a lot of them. So carbon monoxide here binds to complex 4. If we inhibit complex 4, again, we're not going to be able to convert oxygen into water, but we'll still pump some protons. So we can see how all of these are going to have a different impact. Oligomycin is going to inhibit ATP synthase. If we inhibit ATP synthase, we can use NADH, but we're just generating a proton gradient that's not doing anything. We're not getting any energy out of it. And then finally, we also have uncouplers. Uncouplers are a unique protein or molecule. There's a few of them. We're going to look at one example on the next slide. These are going to be molecules that are able to disrupt that proton gradient. So protons get pumped across, and they essentially undo all the hard work of our electron transport chain by bringing the protons back across. So one example is DMP, 2,4-dinitrophenol. So this is a unique molecule, it's amphiphatic. Remember what that terminology means. It has both hydrophobic and hydrophilic properties. This phenol ring makes it hydrophobic. It also has these nitro groups and has a hydroxyl group. Because it's still relatively hydrophobic, it can actually diffuse through the plasma membrane on its own. It doesn't need a transporter. But this is where the uniqueness of that pH on either side of the membrane may come into play or comes into play. This hydroxyl group is an acidic proton. It can be deprotonated. It has a pKa of about 7.2, right around physiological pH. But because of the proton gradients that's generated on either side of this membrane, the pH on either side of this membrane is different. When we look at the cytoplasmic side or the intramembranous space, we see that the pH is typically around 7. So when DMP is on this side of the membrane, that pH now is below its pKa, it's going to allow it to become protonated. Or more commonly, it's going to be protonated. When it's protonated, it can move back across the membrane, and now it's in an environment that has a relatively high pH, around 7.8. Here it's more likely to be deprotonated, releasing that proton and bringing it back across the membrane without the generation of ATP. This is going to be problematic when we want to generate ATP. So this is going to disrupt all that energy and release it. Now this can be problematic because now for a cell, in order to generate enough ATP to maintain homeostasis, it needs to burn even more energy to counteract the effects of an uncoupler such as this. For a while, or in the 1930s, this was actually thought to be a very good potentially weight loss treatment, where essentially cells would not be able to use energy efficiently. It takes burn a lot more energy in order to maintain homeostasis. However, we mentioned that the electron transport chain does not just generate energy, it also generates heat. So in the 1930s, people who were taking this saw a lot of deaths because people were literally overheating. They were not able to maintain their body temperature, had a fever that reached to a level where they ended up dying. So it's been banned since the 1930s for that reason, but even now you can still sometimes see it being used by individuals who are looking for very rapid weight loss. So again, understanding the biochemistry can understand why maybe not such a great idea. So looking at what's happening, We are looking at the pumping of protons across the membrane, but that's not all the thing. We have to account for other things that are occurring as well. In order for ATP synthase to make ATP, it also needs its substrates. It needs ADP and phosphate. We have to be able to get those in the matrix for ATP synthase to carry out its role. ATP is going to be used in the cytoplasm and other organelles. It's going to be hydrolyzed to release that ADP and the phosphate in the cytoplasm. And now we need to bring those into the matrix so ATP synthase can use them to generate more ATP. So that process also costs a proton. So in order to get ATP out and ADP in, we use a translocase that's going to swap them. We also have a phosphate carrier that helps to bring free phosphate into the matrix. So that net process results in also an extra proton coming in. Because of that, we have a net movement of 4 protons into the matrix for every ATP made. ATP synthase brings in 3 protons in order to make 1 ATP, and then we have a 4th proton that's needed to get those substrates in. So for every ATP made in the matrix, we need 4 protons to be pumped out. This can be useful for us when we now go back and look at NADH and FADH too. The electron transport of the redox cycles that are moving those electrons eventually to oxygen are going to pump out protons, and they pump out a specific number of protons. NADH is going to pump out 10 protons, 4 at complex 1, 4 at complex 3, and 2 at complex 4. Because FADH bypasses complex 1, it can only pump out 6 protons. So knowing how many protons are being pumped out by each of these molecules, and knowing how many protons we need to make ATP at ATP synthase, we can calculate how many ATP are made per molecule of NADH and FADH2. And in this case, it's 2.5 ATP for every NADH molecule, and 1.5 ATP for every FADH2 molecule. Now, when you're doing this calculation, half ATPs don't make sense. Always round down if you're when you're doing a calculation later on that's getting you to a full number. There are some exceptions to this. The one that I want you to focus on is that cytoplasmic NADH. Remember from glycolysis? That makes an NADH molecule as well, but it's in the cytoplasm. We need to get that NADH molecule into the matrix where it can be now transfer those electrons to the electron transport chain. In order to do that, we have a series of redox reactions that eventually move that NADH, those electrons, to an FADH2 molecule. The impact, what this means is that the cytoplasmic NADH is only equivalent to 1 FADH2 molecule in the matrix. So we don't get as much ATP from that cytoplasmic NADH. We only get about 1.5 because it's just equivalent to an FADH2 molecule. But there is another transport possible entry point for that to get that NADH into the cytoplasm or to the matrix. We're not going to focus on this course, so it's beyond the scope, but that's the malate aspartate shuttle. There are also some exceptions with the ATP synthase. We do have some isoforms of ATP synthase that are going to require different amounts of protons to generate ATP. Again, in our course, we're going to focus on the one we discussed where 3 protons are required to make 1 ATP. The other thing we need to consider is how much water is formed. Your metabolism generates a lot of water. On average, it's about a cup of water a day is generated in your body just from your metabolic processes. So we can calculate how much water is generated from the breakdown or from this electron transport chain and ATP synthase. In the electron transport chain, the water is coming from that terminal electron acceptor, oxygen, being reduced and turning into a water molecule. So for every 2 electrons that get passed off to oxygen, we generate 1 water molecule. And those electrons are coming from NADH and FADH. So for every one of those that enters the electron transport chain, we generate 1 water molecule in total. At ATP synthase, that condensation reaction between ATP and phosphate is also going to generate a water molecule. So for every ATP made, we generate a water molecule, and NADH can generate 2.5 ATP molecules. So that's going to generate in total 2.5 plus 1, 3.5 water molecules for every NADH oxidized. Likewise for FADH, it's going to be 2.5. 1.5 for every ATP made, for every FADH oxidized, and then 1 water molecule. So we can use this now to figure out what kind of energy and water we're going to get out of the oxidation or the breakdown of glucose in total. So I have a question for you. We have about 5 minutes left. Starting with glucose, calculate the total number of ATPs and waters made through glycolysis, the pyruvate dehydrogenase complex, the TCA cycle, and oxidative phosphorylation. What is the net result of these metabolic processes? In your calculation, remember that you're going to use some ATP at some processes. You're going to use some water at certain points as well, so you need to subtract those from your total. So I'll give you 2 minutes to work through this, and then we'll take it up together on Menti. All right, let's see where everyone's at. Interesting. So a little bit of a spread for those who've responded already. You're getting a lot of 32s, a few 28s, 26s, 30s. I'm going to tell you it is 30. Let's work through that. So looking through glycolysis, let me set this off the screen. Looking through glycolysis, we have one glucose molecule that comes in. That glucose molecule is we need to activate it in the first stage of glycolysis. That's going to cause us to use up two. Let me just do this. We're going to use up 2 ATP molecules. And in the second stage of glycolysis, we're going to generate 4 ATP molecules. So we have a net impact of 2. We're going to generate 2 ATP molecules for every glucose that's going to be broken down to pyruvates. Don't generate any ATP. We will also generate 2 NADH molecules. But remember, glycolysis is happening within the cytoplasm. So those two NADH molecules are going to become 2 FADH molecules in the mitochondria. We're also going to generate some water molecules as well. 2 in one of the steps of the electron transport chain. The pyruvate dehydrogenase complex, we don't generate any ATP, but we do generate a couple NADH molecules with those acetyl-CoAs. In the citric acid cycle, we don't generate any ATP. For those 2 acetyl-CoAs that are moving through, we will generate 2 ATP, 6 NADH molecules. So again, 3 for each time the cycle runs for acetyl-CoA, plus those 2 FADH molecules that succinate dehydrogenase. This process also uses water. So every time the cycle runs, we use 2 water molecules. So in total, we're going to burn 4 water molecules to allow this process to occur. Now in the electron transport chain, we don't have any ATP or GTP generated directly. We will be using up those NADH molecules. So we use up all 8. We're going to have 4, use up all 4. And for every NADH and FADH molecule we use, we're going to generate water. So we have 8 NADH, 4 FADH used. We're going to generate 12 water molecules. Now ATP synthase is where we're going to start to generate some of that energy from that proton gradient. That's going to be coming from the net movement of protons across that membrane from NADH and from FADH. So we use 8 NADH molecules times 2.5, going to equal to 20. 4 FADH plus times 1.5 should be equal to 6. No GTP. We're using up all of those molecules. And then for every ATP made, we're also going to generate an H2O molecule. So 26 should generate 26. So in total, we're going to generate 28 ATP molecules, 2 GTP molecules. We have no net impact of NADH and FADH because everything we generated we used. And then total, 26 plus 12 is 38, plus 2 is 40, minus 4 is 36. Now to make it easier, because my writing and penmanship is not great, I also put this together for you. So feel free to take a picture. I'll throw it up after class, but just so we can get to our last slide, we can look at the sum total now for the complete oxidation of glucose. For every 1 glucose molecule, that's a 6-carbon molecule, we're going to generate 6 carbon dioxides, because it's going to be the complete oxidation of glucose. We're going to generate 28 ATP, but remember those 2 GTP, they're also equivalent to ATP. Those can be interchanged 1-to-1, so we just consider them the same thing.